Hi, this is week 1 of population estimation, and we're going to look at Mark-Recapture. Always got to start with what is a population. It is a group of individuals that are the same species living in the same place at the same time, and they're interbreeding. The population is dynamic, it's growing and shrinking. So we often change this definition based on human factors. In biology, and specifically in ecology, when we're talking about a census, we're usually talking about a systematic recording of every member in a population. So this is something my first-year students frequently have trouble with because in non-biological social sciences, a census is a way to collect information from a human population. It's called a census because it would have been a systematic way of acquiring information about every member of a population. But now it's less so every member of a population. And in biology, it's sort of taken on a different meaning to be, yes, definitely we're counting every individual in that population, or we're learning something about them. And I've just got a picture of a bunch of adorable bee eaters that live in family groups. So we actually can acquire information about every single individual of a population because these guys have very detailed family population dynamics, which are quite interesting. Why would we try to conduct a population census? Why do we even care? This is like one of my favorite questions. We obviously want to know something. Do we want to know how many individuals are in that population? Do we want to know the gender distribution, the age distribution? Generally, if we want to know if a population is healthy or not, we need to know long-term data about that population. We can't just look at this year and say, yeah, that's enough of that population, they're doing okay. Because if we don't have year-to-year information, we don't know if those changes, maybe it looks like there's a lot, but that actually is a tiny amount compared to historical numbers, and that population is about to crash. It's good to have information baseline data so that when things change, we can compare. We're going to look at estimating population size for today. Two main ways of doing this is we can index the population. Most indices involve sampling one point and then extrapolating that to the rest of the population. So for example, I can do bird counts where I stand and listen and look for birds. And then if I know I can hear all the birds within a square kilometer, and this is how many I found, and there's 200 square kilometers, I can make an estimate. This is also really common, even though this is an animal ecology class, this is really common with things that don't move. So you see this in plant ecology, where you do a quadrat and you count how many plants are in a quadrat, and then you estimate that, you extrapolate that to the whole field or forest. It's less common with animals because animals move a lot. So you'll see it where we have groups that are less mobile or are very vocal so we can hear them all. More frequently, we're looking at trying to estimate that population in another way. Mark-Recapture is one of the more common ones. Or you can actually just count all of the animals. Now that we have drones and better aerial photography, some populations we can fly over top of, take photographs, and then literally count every caribou in a herd, every turtle on a beach, every bird nesting on an island. In theory, this is how Mark-Recapture works. If you're in first year environmental science, we use the same formula, but we use slightly different letters. So the N, capital N, is our total population. That's a fairly standard number. Then capital M is our marked population. So we're actually going to take some critters out of that population and mark them. And then we're going to put them back in. Then we're going to take a second sample and we're going to count how many we've captured in that second capture and how many of those are recaptures. If you're in my first year environmental science class, you might recognize these beads. These are my population of fish that I use to do Mark-Recapture in first year environmental science. And as you can see, I already have taken out a portion of these fish and I've marked them orange and I've released them back into the population. I'm now letting them mingle and taking a second sample. So the idea here and what this formula is showing is that the ratio, and wait, let me hold this the other way so it matches the formula. The ratio of marked to unmarked in the total population is going to be relatively the same. It's going to equal the ratio of marked to unmarked in our sample. That's literally what this formula is saying. The total population divided by the number marked in this container equals the total number of fish divided by the number marked in this container. That ratio should be the same if all other things are equal. If births and death rates are even, if immigration and emigration is even, if mingling is truly random, these two ratios of orange to blue should be the same. That's what this formula is showing. There are different ways to do this same Mark-Recapture. If you do it once, where you collect, mark, release at one time, and then go back and recapture once. That's called a Petersen method. And it's the simplest, most cost-effective. It's one of the easiest and most common, but it does rely on one single recapture trying to get enough. Because the more you do this, the larger number of animals you capture, the more likely you get to true representation of the population. So then others will have a multiple collection, continuously marking and recapturing. This is the Schnabel method. So this becomes a little bit more complicated in terms of math because you have organisms coming and going. It's not the marked number is not a stable number. Sorry, I'm showing you this that you can't see. The marked number in the population isn't stable because we're continually marking and recapturing. Then there's some other ones that Dave may or may not talk about in lecture. But for now, the Petersen method is the one that we're going to be looking at. So like I said, we're assuming with this math that everything is staying pretty much the same, that births and deaths and immigration and emigrations are the same. Recruitment is the number of animals born in a year. We're assuming that our marking system is not affecting these animals in any way, that they're still living their lives as normal, that they're not more vulnerable to predation, that they're not going to start behaving differently, and that they'll mix randomly into the group, and that they'll keep their mark, that they're not going to lose it somewhere. Different marking systems that we have are, we get these tiny little metal tags now. They're called pit tags that we can put into critters. Most commonly, this is put into fish. Angling is a huge part of our economy, and there's a lot of research into fish populations because of that economic dependence on fish populations. Bird banding is one of the oldest and most common methods of mark recapture. This is a continuous one where we're continuously marking and recapturing banded birds. And then Dave Hackett has looked at several studies with collars on different larger organisms. When you have smaller organisms, you can actually draw on them. They're too small to tag with some kind of collar or electronic device. So sometimes it's easier to just draw on them. So with a population of ground squirrels or chipmunks, you can actually paint their fur. That paint will stay around long enough for you to do your Mark-Recapture, but then when their coat replenishes the next year, it'll be gone. So it's not long-lasting effects. These snails down here, they've actually added a little bit of cement to their shell so that they can see those numbers. The drawing on the lizard is, again, something that will just leave when they shed their old skin. I'm not sure how the paint on the shells of these little crabs will work, but all interesting ways of marking these individuals. So in our lab that we're going to do this week, we're going to do a Mark-Recapture. You can use beads like this, or in the video that I shared, I use beans. But the idea is we set up a population, we're going to take out some of that population and mark them, replace them, and then we're going to do a series of sampling. Then we're going to do some math to find out what our population size is. So it's important that you don't actually know the number to start off with. Like, I know how many beads are in here because I use the same beads every year for this activity, but I don't want you to know how many there are to start, because I want you to do the math and then see if you actually get a true estimate of the population. So this formula is the same one that we were looking at before. It's still our NM equals CR population, where we have our totals ratio is the same as our samples ratio. We've just moved that M to the other side to isolate total population on its own. And then we've added +1s for error. That's a stats thing. In the lab manual, I've written out a little bit more of an explanation on 95% confidence intervals. So things I want you to consider in this activity, we're going to do 2 different samples, one with larger sampling and one with smaller, to see which is more effective. And then think about the amount of effort it takes. Is it better to take a lot of smaller samples? Is that more effort? Is that more accurate than just a few big samples? Is that less effort? Is that easier? If your sampling that you did was not adequate, how many samples do you think would it take to make a good estimate? What other things do you think are affecting that? Maybe you had a lot of samples that had very few animals, and then other samples that had a lot of animals. Maybe your animals are congregating in patches and they're not evenly distributed. Would that have a huge effect on your sampling? Do you have to violate assumptions to do this? Do you have to stop being 100% random and start making some decisions to get better sampling, quote unquote better? Things to think about. Animals are not random.